All
right. Well, it's my pleasure to introduce Luke Polacek. There we go. From Walloon, Nebraska. And Luke came to us. He's in part of the RHOP medicine program. He's be graduating, of course, on next Saturday with his chemistry health science degree. And in the fall, he will be starting medical school down at UNMC in Omaha. And he's got a very interesting talk that I think is very appropriate for some of the healthcare of the coming future. So, take it away, please, Luke.
Thank you, Dr. Piercy, for the introduction. Just to start off, thank you guys for attending. It means a lot for all of you to be here. Thank you, Dr. Piercy, for being my advisor on this and helping me write a 35-page paper. That was it's a read, that's for sure. So overall, thank you guys for being here. And also, I just want to start off, really, I was interested in this topic by, you're scrolling through Instagram Reels and you get an article that says there are microplastics everywhere. Guess what? That's not really too far off the point, but. So I wanted to kind of get to the root of what's going on there and sort of what health effects microplastics do cause, because they are ubiquitous particles. But overall, my honors project was titled Exposure to Accumulation, a review of micro and nanoplastic interactions with human biological systems. Say that 5 times fast. So to start off, really we're going to be going through what a microplastic particle is, how it enters our body, how it's internalized into our cells, some of the health effects that are caused by that, and then really how we clear and excrete those particles. Starting off, we have to know exactly what are micro nanoplastics. Really, in order to be a micro or nanoplastic, you have to be a plastic first and foremost. Plastics are oil-derived compounds, and most plastics are made up of basically a single monomer, so repeated over and over and over millions of times to make large plastics. So for example, if we have a polystyrene, microplastic or nanoplastic, it's basically a single monomer repeated over and over, and there can be additives in them, but we'll discuss that a little bit later. We're just focused on the monomer point right here. There are also size classifications involved with the, obviously the plastic, microplastic, and nanoplastic. Current literature places basically any plastic above 1 millimeter in size to be a macroplastic or just labeled as a plastic, so that's like a water bottle or something. Microplastics would be anywhere from 1 millimeter then to 100 nanometers, and then nanoplastics 100 nanometers to 10 nanometers. So really that small size paired with the tendency of plastic particles to not break down into their constitutive parts like carbon and hydrogen, and really to break down into smaller polymers of these monomers once again, leads to very small sized plastics in the form of micro and nanoplastics. And we have seen, this isn't really a landmark anything, we do know that cells do internalize micro and nanoplastics, with internalization rates typically going up as those sizes get smaller. So this, I believe right here, was a macrophage ingesting a microplastic particle, and then some evidence on the bottom after it has been internalized by that cell. Also have to discuss the types of micro nanoplastics. Main distinction is primary versus secondary. So primary micro nanoplastics are particles that are generated to be at that size for specific purposes, so like industrial applications. If you need some plastic particles to abrade something, they're going to be generated at that micro or nano size, whereas secondary micro nanoplastics are going to be those that are generated from basically like the sloughing off of plastic particles off of an initial primary plastic. So that could be through something like physical fragmentation. Anytime, if you strike a styrofoam container, it might have something come off, UV degradation, and some chemical degradation as well. We also have many different shapes and sizes of micro nanoplastics, each of which kind of has its own role, but we'll focus on mainly pellets and fibers today. You can have fragments, pellets, films, foams, and fibers. So foam's a little bit of an interesting one. Dr. Piercy, when we were talking about that, that's more of like styrofoam, so like more of an aerosolized sort of plastic, but film, interesting one too. So moving on past that, we also do have to make a distinction between pristine and environmental micro nanoplastics. Pristine micro nanoplastics are plastics that only contain that specific polymer. So going back just one slide, they'll only have, if I have a pristine polystyrene microplastic, they'll only be made up of that. Whereas our environmental ones we see look more something like this, where they have different shapes, different sizes, different colors, and also they have additives with them. Additives are beneficial to add into our plastics to make them more fire retardant, to make them more flexible, and also to make them more hydrophobic and push off water. So overall, those are kind of the main descriptions that we have of micro nanoplastics. So moving into where exactly they're found, on this, I don't want you to focus on the concentrations as much as I just want you to focus on the fact that micro nanoplastics are ubiquitous, especially in aquatic environments. So here they've basically been found in every single water source that we do have. And at this point, we do consider micro nanoplastics to be ubiquitous, not only in aquatic environments, but also in terrestrial, airborne, and subterranean environments. So we are constantly coming in contact with these particles because there's so many plastics in our environment and breaking down in our environment. So from that, it is pretty clear to see that with this constant exposure in the environment, it is going to lead to a rate of internalization and also presence of micronanoplastics within our body. This right here, once again, don't focus on the concentrations as much as the fact that they've been found pretty much everywhere in the human body, in the liver, in the lungs, even in our infants before birth, they've been found, microplastics have been found within them. They've also been found more places than this that this chart even documents. They've been found in the brain, testes, and even artery plaques. So pretty much everywhere, not only environmentally, but in our body, they have been found as well. So really that leads us to the main central questions I wanted to focus on for not only this presentation, but also my paper. How exactly do these micronanoplastics interact with our systems? How do they become internalized, and what are the pathways that allow them to do so? And then lastly, what effect overall do they exert on our health? We're going to start off by describing the entry routes of our micronanoplastics. The main one is ingestion, inhalation, dermal contact, and then injection. Injection is more so, it's kind of off to the side, I don't focus on it. This is like if you need to go to the hospital and you have an IV put in, or if you use injectable medication. So kind of on the side, not as focused on. But ingestion is anything that we eat or drink that goes through our gastrointestinal tract. Inhalation would be anything that we inhale. Some plastic particles can be cleared by mucociliary clearance in the lungs and then go back into the gastrointestinal tract, so secondary exposure there. And also dermal contact through things like normal or broken or damaged skin. We're going to begin with ingestion. In terms of an actual estimation, this article that I abstracted this from is discussed basically in every single micronanoplastic article I've read. You'll probably see this figure over and over again if you do any research on this on your own, but it claims that we have approximately 0.1 to 5 grams of micronanoplastics that we ingest weekly. So that's just ingestion, that's not counting even inhalation. That leads to about 5.2 to 260 grams annually. 260 grams is approximately 2 baseballs, it's 3 decks of cards, and also an iPhone 15 Pro Max. So one lucky winner, look underneath your seat, you just won an iPhone. I'm a college student, I don't have that kind of money. But anyway, so pretty high amount that we're ingesting annually of micronanoplastics. Of course, that's not what we're internalizing, just a figure there. But our food sources of where we get most of our micronanoplastics are things like shellfish and fish, because inherently of the fact that there are many micronanoplastics in the ocean that animals will then eat. It doesn't show it on here as much, but I did see a lot about the clams and mollusks. So if you eat a lot of clams and mollusks, because of their sedentary filter feeding lifestyle, they do have a lot of micronanoplastics. Also, things like food packaging and processing play a role. So that's where more so like salt and honey and sugar and these things that need to undergo so much processing. And also, sorry, Wayne State College, beer has microplastics in it. So processing, the more like machinery it's exposed to, and also packaging, the more physical contact it has with that plastic is going to increase the amount of micronanoplastics present within it. We also do have things like water sources. Between the two, we have tap water and bottled water that are mainly considered for these studies. Tap water exhibits a higher overall minimum, but a lower overall maximum compared to bottled water. Bottled water is going to have more micronanoplastics just inherently because of the fact it's packaged in plastic containers for the most part. So pretty high amount of ingestion just looking at this here. Moving on, we also have inhalation. Inhalation sources are pretty interesting. The main ones that I found were things like polyester clothing. Polyester clothing actually can break down and slough off polyester fibers that are then inhaled, or tire wear. Some synthetic rubbers are petroleum derived and slough off, once again, fibers that can then become aerosolized in the environment. The estimations of inhaled micronanoplastics, though, are pretty variable, to say the least, and I wouldn't really rely on this figure too much, but kind of looking at these deviations, you can see that some of these deviations are as much as the actual figure itself. So things like geographic variation, Omaha's obviously going to have more maybe like pollution because more industries in the area, whereas maybe up here in Wayne we won't have as much micronanoplastics present in the air. So things like geographic variation do need to be taken into account, which is something that unfortunately really isn't seen in current research as much. But in terms of size limits, also important to mention a size limit of inhalation. In order to actually reach the alveoli, micronanoplastic particles have to be at a size less than 5 micrometers. Those that are larger than that typically tend to be cleared by mucociliary clearance. This is where micronanoplastics get caught in the mucus and get pushed upward into the pharynx by ciliated cells, where it can either then be expectorated or spit out or swallowed through, and then secondarily exposed to the gastrointestinal tract. Or it can be deposited, causing some issues in deep lung tissue. Moving on from there, we do have dermal contact as well. Really, I'm not going to talk too much on this. It's the least concerning exposure route, and really only nanoplastics that are less than 100 nanometers can cross this epidermis layer to access lower dermis layers that have more circulatory involvement in them. So really not too concerning. If you do have like damaged skin, however, like pretty deep cut, or something like maybe you have like really dry hands so they crack really easily, could introduce you to some microplastics, but really it's not even considered in most articles to be a significant source of micronanoplastics. Sources, however, are things like personal hygiene products, so toothpaste, shampoos, makeup, lotion. That doesn't mean to stop using things like deodorant and toothpaste. You'll probably have a bigger problem if you stop using those things, but there are some micronanoplastics. And this image, interestingly enough, was from an article discussing Crest toothpaste, and they found that these little beads, some of these are actually micronanoplastics. So watch out if you're using Crest. Sensodyne's the way to go. Really, there's a common theme with at least our exposure here, and it's that the lifestyle factors do shape our exposure to these particles. This graph right here, on the y-axis, you can kind of see it says microplastics concentration items per grams of BW. Guess what? That's dried feces. So what a great thing to put on the y-axis, but this is the amount of microplastics that they found within individuals' feces. So things like takeout food, and really like these two groups right here, we'll talk a little bit more about this later, but these are individuals with inflammatory bowel disease, so breakdown of some of that lining in the GIT. And then up here is just healthy individuals, but really in both groups we found that takeout food, as you eat more and more takeout food, you have an increase in the amount of microplastic particles that are present within your feces. Same thing goes for bottled water. The more bottled water you drink, there's a significant difference between these groups in terms of how many micronanoplastics. So probably good to focus on things like lifestyle factors that will go ahead and tell us how much an individual will be exposed to. Now we move into more of the section that's based on how we cross some of these barriers and more of the mechanisms. For the gastrointestinal tract, passing the gut epithelium specifically, there is a maximum size limit considering all of our different mechanisms of about 150 micrometers. So anything above that simply moves through the gastrointestinal tract and then is expelled via the feces. But mechanisms, really I do want to say though before I continue, the field of like micronanoplastic research is very novel. So a lot of the research that I'm going through are articles that are past 2020. Like this is a 2023 article, and this was the most recent, well really like the most detailed one I could find regarding mechanisms of how they actually travel through. First one would be transcytosis though. This is where once micronanoplastics kind of cross this mucus layer, the GIT, they're able to be endocytosed by gut epithelium and then expelled on the opposite side, which then makes these particles available to things like lymphatics and macrophages, and then also circulation. This image right here, you can see just a little bit on the side how there is some circulatory involvement past the initial layer of epithelium. There's also internalization via M cells. M cells are immune cells present within the Peyer's patches region of the ileum. These cells do play a role in things like basically just the immune system of the gastrointestinal tract, so they can also do something similar to transcytosis, where they will internalize these particles, expel them past the gut epithelium, making them available to lymphatics and circulation once again. The last mechanism is probably the least major, I would say. It's paracellular transport. This is where small enough particles are able to move in between cells through their gap junctions. You have to be extremely small to do this, or the integrity of the GIT has to already be compromised. So really not as major of a route. But overall bioavailability figures of initial doses show about 0.1% to 1% for 20 micrometer polystyrene plastic particles. This was a mouse study, so not within humans. We can't do human studies on micronanoplastics, a little bit unethical there. Next up, we have the alveolar membrane itself. The particle size limit in this case is anything above 5 micrometers to even reach that alveolar membrane. With the exception of fibers, some fibers can kind of like reach in and become deposited, causing some damage within that area. The mechanisms here are pretty similar though. We have transcytosis, which is once again where cells will internalize and then go ahead and exocytose them on the opposite side, making them available to once again circulation, lymphatics as well. We also in the lungs have macrophages, so endocytosis by macrophages may occur, going then to lymphatic drainage, which is then internalization, and then paracellular transport in an identical manner to that first one. Bioavailability of this is about the same as the first. It's 1% for 3 micrometer polystyrene. plastic particles once again in mice. So now we're going to move on to the section of this where we talk about some of the neurological effects of micronanoplastics. First thing I want to say is that there are pretty limited human studies on micronanoplastics within the central nervous system, and they're mainly behavioral and things like mice. So they'll test like maze tests, or they'll do anxiety tests on mice to see how it affects their behavior and overall like social behaviors aspect of that as well. But I want to talk about this article specifically. This is an article that focused on introducing mice to varying levels of microplastics, and then also introducing vitamin E, which is an antioxidant. They focus really heavily on the role of reactive oxygen species, so agents of inflammation and how micronanoplastics may exert their effects. But with that, they did in this study find things like learning, memory, and behavioral deficits within the mice, and then really wanted to look at what's going on with these different groups. So focusing on these, like the x-axis on all of these, we have saline, which would be our control group, 0.01 milligrams a day of polystyrene, 0.11, and then adding 1 milligram plus some of the vitamin E, then a vitamin E group by itself. With this specific study, on each y-axis we have ROS, or those reactive oxygen species, which are indicative of inflammation, MDA, which for our intents and purposes we're going to consider pretty similar to this, and then GSH, that's reduced glutathione. It's an enzyme that plays a role in inflammation. So we would expect low inflammation to see high amounts of reduced glutathione, and then high inflammation, kind of low levels of reduced glutathione. With this, as they increase the concentration, you can see that there are significant differences in the amount of reactive oxygen species that do form. Same thing with reduced glutathione, we decrease overall with these higher concentrations. Interestingly enough, you can kind of see somewhat of a rebound when they add in vitamin E, the antioxidant, to these studies, but they really noted that there was a big behavioral difference overall when they added in an antioxidant to these studies. So perhaps reactive oxygen species are the manner in which this micronanoplastics affect the neurological system in this case. There's also one of two studies I was able to find actually on human neuron cells. T98G cerebral cells, they exposed them to various levels of polyethylene and polystyrene, so they found that there was a significant amount of reactive oxygen species formation with those really high concentrations again. Moving on to cardiovascular effects, we have found that there are micronanoplastics in our bloodstream. This study that I quote right here is a landmark study from 2022 that was really the first to show that we have these particles in our bloodstream. Past that, because of the fact that they're in our blood, they can do things like accumulate within plaques. This is taken from an article where individuals underwent an operation to remove plaque from their carotid arteries. Once it was removed, they went ahead and analyzed it and found that there were microplastics present in those atheromas specifically. Because it's in the bloodstream, it can also lead to alterations of blood clotting and increases of certain inflammatory proteins. I told you, Dr. P, right there, IL-6. So with blood clotting, this is a study from South Korea focusing on the levels of C-reactive protein in the blood, which is associated with the innate immune system and inflammation, and also fibrinogen, which is a clotting protein. But really the important one to talk about is right here, it's APTT. A higher APTT indicates a higher amount of time that it takes to clot. So individuals with low microplastic counts in their blood clotted their blood significantly faster with a p-value of 0.037, that is, than those with a high concentration. Additionally, they did find correlations with C-reactive proteins, so the more, not super strong correlations to be completely honest, I don't think they found significance in this, but they wanted to mention it. Increase in C-reactive protein here, and then also increases of fibrinogen. So fibrinogen going up, it's a clotting protein, but then also their clotting time going up. It's a bit paradoxical, but they kind of mentioned that as well in their findings, that it was a bit strange to see that. But when it comes to some of these inflammatory proteins, I'm talking about cytokines, so things that help to proliferate some of this inflammation further. We have IL-6, which was found to be significantly increased in some of those higher micronanoplastic groups. So IL-6 is associated with the acute immune reaction, which leads to increased inflammation, and overall, it's an innate immune response. Do I get 100%? Yes. Perfect. And also kind of more of like a non-discriminatory effect. We have hemolysis of red blood cells at very high concentrations of micronanoplastics. We also do have gastrointestinal and microbiome effects that are present with micronanoplastics. Interestingly enough, I did find multiple studies that did show that there are significant increases of micronanoplastic concentrations of individuals, specifically For inflammatory bowel disease, there was an increase overall in the amount of micronanoplastics in their feces, so pretty interesting point there, but also liver cirrhosis, higher overall amount of micronanoplastics within individuals with liver cirrhosis in their livers. But the main way in which that micronanoplastics do interact with our gastrointestinal tract is through altering bacterial populations just due to the presence of those plastics. With specifically, I believe it says polyethylene, PCl, and PLA right there, we do see changes in bacterial populations, like an increase in the phylum Firmicutes and a decrease in the phylum Bacteroides. I hope I'm pronouncing that right, but overall, the presence of micronanoplastics does change bacterial populations. And really what that does is it doesn't just change the microbiome, but it also changes things like the metabolites that are present within your microbiome and gastrointestinal tract. One of the most important ones that I read about was shorty chain fatty acid production. That is associated with these changes specifically in the gastrointestinal tract. We see an overall decrease in shorty chain fatty acid production. Normally, shorty chain fatty acids will kind of work to not completely suppress the immune system, but suppress it a little bit so that it doesn't attack the microbiome, but then also plays a role in things like mucus production. So mucus production helps to maintain the integrity of the gastrointestinal tract, and when we decrease that, it kind of decreases that integrity as well. And then also inflammation that can be caused from this lack of immunosuppressive action can cause further damage. So I did think it was interesting to see a parallel between something like inflammatory bowel disease, which is associated with gastrointestinal inflammation, and some of these effects that micronanoplastics are causing, like a decrease in integrity and inflammation. So could be correlated. I don't know. That was interesting. Moving on past that, chemical additives within plastics do hold their own potential for damage. The use of additives, I kind of already discussed that, was things like fire retardants and to make plastics more flexible or also more like hydrophobic so that water doesn't stick to them. But really, most of these additives that we're using, things like bisphenol A or our phthalates or our PFAs, are also endocrine disrupting chemicals. They interact with different receptors in our endocrine system, like thyroid receptors, progesterone, estrogen receptors, a lot of different things associated with our endocrine system that overall lead to changes like, in men, a decrease in sperm count and quality, in women, a decrease in oocyte quality, and also some issues with birth complications. The main one I did see was a lower overall gestational age because more women are having premature births as a result of some of these additives. And with BPA specifically, it's interesting. It's been found, this was a study analyzing Americans' urine, that's kind of a weird sentence, but 92% of Americans did have BPA within their urine, indicating that sort of a chronic level of exposure in our environment. For both sexes, though, it causes metabolic issues and cancer overall. So lots of issues associated with these endocrine disrupting chemicals. And overall, really kind of like looking at the facts here, additives may be more harmful than micronanoplastics may be alone. So having additives within our micronanoplastics may lead to an overall sort of like combination effect where it's more dangerous. Really, that kind of is a cause for alarm, just a quick aside, because most of our studies use pristine micronanoplastics instead of using realistic environmental micronanoplastics that contain things like BPA or phthalates and all these other additives. Past that though, looking at some of our persistence and excretion mechanisms, really we could have short-term versus long-term persistence of these particles. And overall, out of everything I've talked through so far, this is the most unknown. We really don't know a lot about how these particles are cleared from our organs, our bloodstream a little bit, but from like our organs more specifically, we really don't have that great of an understanding because it's such a novel field of research. But short-term persistence is believed to predominate over long-term. So short-term, plastic particle may become internalized, but probably isn't there for years or something like that. Excretion mechanisms, though, could be simple movement through that gastrointestinal tract. We already discussed a few slides back how there's that size limit of 150 micrometers. If it's above that, or even if it just doesn't get internalized, it'll just simply move through and be expelled in the feces. Past that, we can also have removal of micronanoplastics by the liver, so this could be circulation. Two major routes that I did see were that liver macrophage cells, these Kupffer cells right here, will internalize microplastic particles, and then that'll at least clear them from the circulatory system, but from there's not much known about how they get rid of them from the Kupffer cells. There's also hepatocytes. Hepatocytes can also ingest these micronanoplastics and actually reinsert them into the bile, which will then reintroduce them into our gastrointestinal tract, leading to some sort of like secondary exposure, for lack of a better term. And lastly, there has been micronanoplastics found within our urine. We don't know the mechanism though as to how they really enter our urine. Probably not through the glomerulus, because this has a filtration size limit of about like 10 nanometers, and we are finding micronanoplastics present in the micrometer size. So it is thought, there was a theory in one of the articles I did read regarding this, that perhaps instead of like becoming inserted within the nephron system here, micronanoplastics may become associated with that urinary tissue through other sort of blood vessels, but really not a good theory, not any good theory about how it gets out via the urine. But kind of overall, looking at everything we talked about, we have talked already about how micronanoplastics, we're constantly exposed to them. They're ubiquitous within our environment because of the increase in plastics and the breakdown and overall durability of those plastics that we do have. They also have indicated some ability, at least, to affect our health, not only through just the simple presence of them alone, which we've talked about for most of this, but also through the additives present within them, those endocrine disrupting chemicals. Moving past that, we do believe that excretion occurs, otherwise we'd have, like micronanoplastic tumors or something like that. But short-term persistence does dominate out of this, or at least the current research does indicate that short-term persistence is the current really route. But overall, looking at all the research that I've done and really reading all the articles that I have, I do believe that we should consider micronanoplastics as being possible emerging contaminants. And again, we don't know for sure if they have clinical relevance or correlation to some of these increases in maybe chronic diseases we're seeing, but it is important to keep in mind that we are seeing increases in micronanoplastics in our environment and to be aware that they could have possible clinical relevance. So with that, I just want to say thank you guys for showing up. Thank you again to Dr. Piercy, being my advisor on this. I really appreciate your work with me. And I also do have my references in my paper available upon request. So if you want to read my paper, once again, 35 pages to read, but it goes through a lot of what I talked about. There's a lot I had to cut. Really wish I could include it all, but otherwise I'd be talking for an hour and a half. So anyways, thank you all for coming. Really appreciate it.